Jeff.
Thank you.
Jeff, you see what you want here.
To first thank Miguel and also the other organizers fighting me. I'm an engineer, and so over the past decades at Hopkins, my lab, we are interested in working on the development of new technology, molecular analysis of diseases, and for cancer, but also for other diseases such as infectious diseases. So I'm going to share with you some of the examples of using the nanotechnology to enhance detection of DNA methylation, circulating DNA.
Yes, we did do that. So you see, it says one-time charges, negative $10.
So DNA methylation contributes to the progression of tumorigenesis by silencing of tumor suppressor genes. I have found that, and our report shows that the promoter regions of DNA from normal cells is often unmethylated. And the promoter of a tumor suppressor gene coming from tumor cells often been found to be hypermethylated. So this suggests that, in fact, people have been using this as a marker for cancer. But more importantly, a lot of recent reports shows that the DNA methylation events and can be found in early stage or even precursors such as polyp for colon cancer and also penny for pancreatic cancer. Highlighting the promise of using DNA methylation as a marker for detection. Currently, one of the most commonly used methods for promoter DNA methylation detection is MSP methylation-specific PCR, first developed by Jim Herman and my colleagues and Steve Baden at Hopkins. The methods and other related methods, they have typically high sensitivity and specificity. But many of you here know there's also discussion here that PCR-based method, including MSP, the sensitivity is still not high enough to be used to reliably detect the circulating DNA or tumor DNA. And has to use nested PCR or even digital PCR enhance the sensitivity as well as resolution. The MS methods as well as other methods such as bisulfite sequencing also have some problems with that we just discussed, involved a very tedious process of sample preparation, including DNA extraction, bisulfite conversion. So current methods to process these samples has unfavorable yield in terms of recovering the low abundance of DNA from the liquid biopsy and also do the processing. So the lab has been working together with several labs at Hopkins, including Jim Herman and Steve Baden's lab, try to improve the detection and also to improve the sample preparation for DNA methylation. I'll give you some examples in the past few years. First example that I'm going to share with you is that we are proposed to use the quantum dot rate technology to enhance the detection of methylation using MSP. So we have this technology called MSQ-rate. This method that we first conduct the bisulfite conversion to convert unmethylated citizen to become uracil. So that we're using the PCR primer to amplify the methylated targets with a limit cycles, somewhere about 10 to 12 cycles. And we add quantum dot rate sensor to detect this methylated DNA. I'm going to give you a little background on this DNA, quantum dot rate DNA nanosensor technology that we developed about a decade ago. So this DNA quantum dot nanosensors has three elements: a pair of the hybridization probes. One labels with a Cy5 dye serve as energy acceptor. Quantum dot serve as an energy donor. So the probe bind to the target DNA, and the whole thing will be captured by the quantum dot through the strip actin biotin. And then we excite the quantum dot with certain level of energy. A part of the energy will be transferred to the Cy5 and then through the rate. And then we check this one, we can confirm the presence of the DNA and also quantify. So many of you have known that the quantum dot has several unique physical optical properties, very low emission band, very large stock shift, and also high quantum yield. So this allows us to be able to detect this quantum dot nano that has very low background noise. But upon binding to the DNA, it can emit a high signal, and this signal can be detected at a single particle or single molecule level using this quantum dot technology. And couple this quantum dot free technology with MSP, and we are able to quantify the DNA methylation according to the level of rate. Quantum dot signal decrease and energy acceptor signal increase. So this can be used to quantify the DNA methylation level. And we also demonstrate that with this quantum dot enhanced detection, the sensitivity of MSQ free now allows us to be able to detect around a few tens or about 10 or 15 picograms of the DNA, which is about a few copies of genomic equivalent, but in a very high access of unmethylated DNA background. In this case, 10,000 access of the background DNA. We demonstrate that this technology not only can be used to detect the serine sample, but also can analyze some other biological samples such as sputum. So here we demonstrate the use of this quantum technology to detect the methylation in sputum sample and compare the technology with gold standard, one of the commonly used method, MSP method here. So for the 20 samples that we detect, MSP did not detect any methylation signal. Only when we conducted the two-stage nested MSP, we started to see that 3 samples, 3, 7, and 8, that shows the methylation for PCY cut here. And using this quantum dot methods that we can detect you see patients that samples contain methylation, but without using this nested amplification steps and give very high signal to background ratio. But the quantum dot enhanced MSP, other MSP, also real-time methods still have some limitation that this method cannot be used to analyze heterogeneity of a polymer methylation. So recently, we proposed an alternative approach we call streaming technology. So using technology, we combine the digital detection, digital PCR concept together with a Miller curve analysis, try to analyze individual copies of epiallelic species with at a single CPG site resolution. So the general concept of this streaming assay is that we first performed limited dilution of the samples, so that each reaction well contains no more than 2 epiallelic species. Following PCI and also Miller curve generation. So for the microwell contains only the unmethylated DNA will shows up for peak. But for the well containing the fully methylated DNA or hemi-methylated DNA will show a secondary peaks with different melting temperature. And this temperature can be used to discriminate epiallelic variants based on the methylation density. For the DNA methylation, the first step is to conduct bisulfite conversion. So bisulfite conversion can convert the unmethylated serine to be uracil. So after PCR, the amplicants from methylated DNA with a low GC contents. The G content level increase with the number of the methylated CG sites here. So the melt temperature to detect the different alleles variants and also to quantify the methylation density. Here shows that this is one of the experiments that we mixed a small quantity of 2 different variants. One contains 20% of 120% of the methylation density, and the other variant is 100%. In a very high excess of unmethylated DNA background, in this case 50,000 high excess of unmethylated DNA background here. So we expect to detect a 4.2% curation, 4.2 copies for 20% variants, and also 4.2 copies for this fully methylated variants. Streaming assay reports detection of 3 copies and also for 20% and 5 copies for 100%. And this data shows that the analysis to analyze the heterogeneity of the DNA methylation is BRCA1 gene from the patient blood samples here, and you can see they are with the effect is quite variable. It's not the same. And for this patient one here, that it has only 1.7% of epiallelic fractions, And the technology is even be able to detect the epiadic fractions down to 0.05% as the case here.
Okay.
So the method that I'm here currently is conducted using 96 or 384 plates. So this setting has a limited range, and also that's why we have to do limited dilution. And throughput is also limited. So we are working on the development of microfluidic reaction chambers to enhance the throughput and also dynamic range. So in this case, we have this device here has four regions of 4,000 wells. Within each well, we can conduct the action and also Mel curve analysis. We are hoping that this microfluidic coupling with this microfluidic LH can further enhance the streaming technology to tackle the more challenging analysis of challenging clinical samples. So we just talked in the pre-talk from David talking about the importance of a sample preparation. We know that for the DNA methylation in particular involve very tedious sample preparation process. Not only you have to extract DNA from the body fluid, from the blood, serum plasma, and you also need to conduct bisulfite conversion. So this process also involved many laboratory steps. Importantly, many transfers of DNA between different. So this responsible transfer cause loss of DNA and also compromise the sensitivity. Okay. So developed a protocol method we call methylation on beads or MAP. These methods. So in this method we use silicon paramagnetic particles as a single carrier to carry the DNA away from every different steps and to make this be a single tube process to minimize the DNA loss during the set DNA extraction as well as bisulfite conversion. So it shows that we compare the sample preparation method, the MAP, to the conventional method here. So this is the patient serum samples from 20 patients, from 15 patients here. So we see a consistent enhance in terms of the yield of DNA recovery from the blood and also bisulfite conversion, average 6.7-fold of enhancements based on this study. Also show that this method is applicable to other sample types, including sputum here. So more recently, my colleagues Nida Huja at Hopkins reported find that two genes, the PC1 and ADAM1, these two genes, not only they can be detected in pancreatic tumor, not only at the early stage, but also can be found in the penny, which is the precursor for pancreatic cancer. We then move on to see whether or not we can detect these two novel genes in blood using this nanotechnology assets. So we conducted a small cohort studies that we test 42 patient samples, cancer patient samples plus 36 normal patient samples. We find these two give us about 85% and also 85% sensitivity specificity about this range. But very interesting that we find that for the early stage the patients that in the serum samples we can detect 9 out of 10 of BNC and also 9 out of 10 from this beta from the ADAM this gene. So this is pretty promising and encouraging results for us. Show you that we have developed this streamlined method called methylation on beads, be able to streamline the whole process into a single tube. So also motivated us to develop technology that we can using this MAP protocol and develop microfluidic technology that we can integrate sample preparation by DNA extraction by bisulfite conversion to get DNA all on a single device for high throughput, more robust analysis that is needed by routine clinical utilization potentially for point of care. So I'll show you one example. It earlier works that we are working on a digital microfluidic technology where we're using droplets to sample. So this is the standard process for PCR detection, beginning with lysis and solid-phase DNA extraction using columns or silica beads and water dilution, and then purify DNA substrates through PCR. So what I'm going to show you in the next two slides is that we are able to miniaturize this whole thing, microfluidic devices in a droplet format. So on our platform is that we use this drop as a storage of all necessary buffers, including lysis buffer, binding buffer, elution buffer, and PCR buffer here. And then the idea is that once we dispense the sample to mix with lysis buffer, DNA got released, and then the beads, magnetic beads will capture this DNA. Next dropped, and this is wash buffer, and another wash buffer for wash. So this reverse the process doing the buffer exchange when you do the bench process using this tube-based preparation. So I'll show you this video in the whole concept. So is when you dispense the sample to mix with the lysis buffer, released and captured by the particles and be to lysis the wash buffer, depending on what kind of reagent collagen or different manufacturer used. And then purified DNA will be subject to elution and bring to the chamber here to the PCR. So everything is covered by oil, so this do inversion PCR. So we can do the direct the PCR measurements right on this droplet. Can do this here in a manner. And then we also have recently, we also demonstrate that the bisulfide, the second stage, the bisulfide conversion chemistry. This also tedious process also can be done using a similar concept using drops. So also the instrumentation also can be quite is that we're using electromagnets. This is basically very small electromagnets printing on coils. Printed circuit boards, very small one, and then you can apply the currents to the coil, you generate magnetic fields. If you apply currents to adjacent coils with different polarity, we have to create magnetic gradients. So these magnetic gradients allow us to move magnetic particles together with the drops, coil-induced magnetic fields and magnetic gradients. And so, it's also important to do a mixer. We also show that if we switch between the polarity of adjacent coils, we are able also to do the mixing at this micro droplet scale. So you can do this efficient mixing using by switching the polarity between the coils. And, missing what is this? Some of this is the slide showing that this is a magnet. That is the entire process integrated with sample preparation and also downstream is PCR process. That is buffer, and then beads will and go through first wash buffer here, and you see still some residual composition there, and that's why you have to go to multiple, typically 2 or 3 wash steps. Typical simple preparation depends on the chemicals you use, and you will carry this purified DNA agitation, mixing, and then downstream. And then when you go down to this PCR, again, the device is covered with, so we can do immersion PCR right on this device. So for this, I would like just to conclude my talk by thanks my collaborators, Jim Herman, Steve Bailey, we have been working for 10 years, Malcolm and also students involved in this project, as well as funding agents, especially NCI. Thank you.
Question, Jeff?
How far have you pushed the row of detectable methylated DNA to methylated DNA? I see it's something like 1 to 15,000. Have you pushed that envelope to see how far go with this?
The result I just showed is 1 to 50,000.
Right.
But that is for analytical validation, because from real clinical, you really don't know what's the actual ratio. So we can push further far with microfluid density, with 84 and
right.
do limiting dilution, so it's really not an ideal case. So with this mega array, the one now I show you is like 4 times 4,000. So we are making this mega array, so we can go on higher one. But real clinical utilization really depends on the real patient sample. So clinical validation, that to answer your question, that now we can do 1 to 50,000 and can be pushed further down with the microfluidic arrays.
Okay. Thanks, Jeff. Next speaker is going to be